Bismillahirrahmanirrahim. Assalamu alaikum. This is FAHMIDA CHANNA, lecturer Zoology. Today I am going to deliver a lecture about enzyme. What is enzyme? Enzyme is a Greek word. It means in, enzyme means yeast. Its meaning is a misnomer to its name. Enzymes are biocatalyst molecules. What do you mean by biocatalyst? Bio means life, catalyst means those molecules which speed up the chemical reactions is known as catalyst. Enzymes are those which are increase or speed up the biochemical reactions. Discovery of history of enzyme. Friedrich Wilhelm 1878 named biocatalyst as enzyme. Thomas Cash and Sidney Altman during 1980 discovered that some ribonucleic acids also behave as enzyme. Now therefore that is known as ribozymes, because most of the enzymes are the proteinaceous, but some of them are the ribozymes. Properties of enzymes. Now let's talk about that. Generally, as just I told you about, enzymes are proteinaceous. It means most of the enzymes made up of protein. They react with both acid and base due to protein in them. They are specific in their nature and their action. The enzyme molecules are much greater in size than their substrate. It is a very important point. Enzyme molecules are always larger than their substrate, as showing this figure also. They are particular site to react with the substrate, that is known as active site or particular site. At that site, any particular substrate is attached with enzyme. Enzymes are required in a very small quantity. Enzymes remain unchanged before and after a chemical reaction. That is a very important point for the MCQs. Enzymes are biocatalysts, as just told you before. They are speed up the rate of reaction. They are heat sensitive. It means they are the thermoliable molecules. If heat is increased or decrease, enzymes activity is also affected. Enzymes activity can be accelerated by certain ions, that is known as activators. It means those molecules which activate the enzyme's activity, that is known as activators. These are, most of them are the metals like manganese, nickel, magnesium, chlorine, etc. Their symbol is showing. Enzymes activate can be inhibited by certain molecules or factors, that is known as inhibitors, like DDT, parathyroid. Examples of the concentration of enzymes activity like a substrate concentration, enzyme concentration, pH, temperature, etc. Now let's move to talk about the classification enzyme. Classification basis of site of reaction or action. On the basis of site of action, enzymes are two basic types, endozymes and exozymes. Endozymes is also known as intracellular enzymes. Endo means in, it means intra. Zyme means enzymes. They are act within the cell. They are secrete from that and act on it. That is known as endozymes. Exozyme enzymes are those they are act outside the cell. Therefore, that is known as extracellular enzymes. Most of the digestive enzymes are extracellular enzymes like pepsin, ptyalin, etc. Now, classification of enzyme on the basis of the structure. On the basis of structure, composition of enzymes are divided in two basic categories: simple enzymes, conjugated enzymes. Simple enzymes, it means those enzymes which are only made up of proteins, that is known as simple enzymes. Conjugated enzymes. What is conjugated enzyme? Conjugated means union. Conjugation means union of two different things. The conjugated enzymes are those which made up of two different parts, apo part and prosthetic part. Apoenzyme or apo part, the proteinaceous part of conjugated enzyme. It means that part of enzyme which is only made up of protein, that is known as apo part or apoenzyme. Prosthetic group, it means non-proteinaceous part of the conjugated enzyme. Conjugated enzyme, also known as holoenzyme, when they are show full range of activity. That Now let us talk about the cofactors or helper molecules. These are the non-protein part are chemically compound required by enzymes to catalyze the certain chemical reactions. Common cofactors are metals, as just told you before that, manganese, nickel, magnesium, etc. Types of cofactors, coenzyme. If cofactor is organic molecule, that is known as coenzyme. Activators, as just told you before that, those which are activate the enzyme activity. Inhibitors, that is cofactors that are inhibit the enzyme activity. Now the types of inhibitors: competitive inhibitors, non-competitive inhibitors. Competitive inhibitors are those cofactors that bind at active site and inhibit the enzyme activity, as showing in this figure also. Non-competitive inhibitors are those which are bind at allocentric site, it means other side than the active site. They are bind it and inhibit enzyme activity, that is known as non-competitive inhibitors. Now let's to test evaluation for the students. Kindly read it carefully, the all MCQs, and solve it. Please share your feedback or comments. Thanks to watching this video. In the next lecture, we will discuss about other topics related with enzymes and complete the chapter of enzymes. Allah Hafiz.